And it's my pleasure to introduce our next speaker, who's Julien Bryois. He has studied in Geneva and is based now at the Karolinska working with Pat Sullivan on expression data to follow up GWAS signals. The floor is all yours, Julien.
Thank you very much, and thanks for the invitation. So, as you know, over the last 10 years, we've made significant progress in understanding the genetics of complex brain disorders. And through GWAS, we have found more and more loci associated with different brain complex traits. And so these studies are great in mapping SNPs to disease, but they don't tell you how the SNPs are connected to the disease. And this is key if we want to develop new therapeutics, because we need to understand exactly what is the causal biology of brain complex traits. So this is challenging for multiple different reasons. The first one is we often don't know which are the causal variants because of the correlation structure of the human genome. We often don't know what are the risk genes because most non-coding variants are located in, most associated variants are located in non-coding regions of the genome. And we don't know in which cell type those loci are active. And this is probably key, in my opinion, to develop functional genomic assays in the right relevant cellular context. So the question was, well, which cell type are relevant? So as you know, the brain is a very complex tissue with lots of different cell types. You have neurons, oligodendrocytes, astrocytes, vascular cells, microglia, and within those broad categories of cell types, you can have different types of excitatory neurons or inhibitory neurons. So our strategy to find out which cell types are relevant to brain complex traits has been to integrate GWAS summary statistics with single-cell RNA-seq. So a few words about single-cell RNA-seq. So what you do is you take your tissue of interest, in our case, the brain, you dissociate the tissue into single cells, generate gene expression profile for each single cells, and then you apply clustering algorithm to identify cell type. So what you could get is something like that, where all the different single cells that are neurons cluster together, and you can label them based on the genes that are expressed in that cluster, and same for astrocytes, oligodendrocytes, etc. So basically, it's a way to recapitulate and find out all the different cell types in your region of interest. So then, or you can of course average all those different single cells to get a pure, essentially pure gene expression profile for your cell type. So this is essentially what you can get. So three different genes, many different cell types. And so you can see ApoE, which is associated with Alzheimer's disease, and you can see very strong expression in microglia in astrocytes. Here, the dopamine 2 receptor, which is the target of antipsychotics, which are used to treat schizophrenic patients. And you can see expression in dopaminergic adult neurons, hypothalamic dopaminergic neurons, and medium spiny neurons, and PCAM1, which is a marker for endothelial cells. So we needed to move from that expression space to a measure of how specific the gene is to the different cell types. So we did something very simple. We just divide the expression of each gene in each cell type by the total expression of the gene in all the different cell types. So you can see the picture is exactly the same, but now all the genes are on the same scale from 0 to 1. And so what this means is that, for example, 45% of the total expression of ApoE is performed in microglia and 35% in astrocytes. So from that, we essentially can find what are the genes that are specific to the different cell types in the brain. So we can test we can get the 10% most specific genes for microglia, astrocyte, endothelial cells, pyramidal neurons. And the idea is that if the cell type is not relevant to the disease, those very specific genes should not be enriched in genic association with any brain complex traits. While if the cell type is very relevant, then those very specific genes should be enriched in genic associations. So we can test that using two different methods, LD-square regression and MAGMA, which provides evidence for heritability enrichment or gene level enrichment in genic association with different traits. So we published two years ago our paper on schizophrenia, where we found association with pyramidal neurons from the somatosensory cortex, pyramidal neurons from the CA1 region of the hippocampus, medium spiny neurons from the striatum, and cortical interneurons. But this was only taking advantage of 24 cell types from 5 brain regions and using 10,000 single cells. So this was, I think, a great first pass, but of course the brain is more complex, there's more brain regions and there's more cell types. So we thought at the end of the paper, we thought, well, what about the other cell types? Maybe there's other neurons that are more important than the pyramidal neurons in the cortex and hippocampus. And then what about other traits? We focused on schizophrenia because the genetics was the most advanced, but in the meantime, we've seen larger and larger GWAS for other brain disorders. So in 2018, our colleagues at Karolinska did these amazing data sets where they sequenced more than 500,000 single cells in mouse from 19 regions of the nervous system, which allowed them to identify 39 broad categories of cell types. So they sampled pretty much everything: the cortex, the striatum, amygdala, hippocampus, midbrain, cerebellum, and also the peripheral nervous system and the enteric nervous system. So first we wanted to see if the method was working for all the different traits using tissue level gene expression data. So we used human RNA-seq bulk data from the GTEx consortium and tested 26 different traits, 18 brain related and 8 non-brain related. And so what you can see are the strengths of the association on the x-axis for different traits. And the bars are colored in orange if it's significant at 5% FDR using both magma and L-score regression, or in blue if it's only significant at 5% FDR using magma. And so you can see for schizophrenia, we saw no enrichment in any tissue that is not brain related, and the strongest signal in the brain cortex. For Parkinson's disease, we found the strongest enrichment in the substantia nigra, which is the expected region, because that's where dopaminergic neurons that are characteristically lost in the disease are located. For type 2 diabetes, we had the strongest signal in the pancreas, and for stroke in blood vessels. So this was reassuring and suggests that the findings at the cell type level are meaningful to find which are the relevant cell type for the different brain complex traits. So we mapped cell types from the entire nervous system to 18 brain complex traits, but in the interest of time, I'm only going to discuss schizophrenia and Parkinson's disease. So the results for schizophrenia are the following. We found at the top telencephalon projecting excited neurons, telencephalon projecting inhibitory neurons, and inhibitory interneurons. And these are essentially the same cell types that we had found 2 years ago. So the projecting excited neurons group is a broad cluster that includes pyramidal neurons from the cortex, the hippocampus, and from the amygdala, while the projecting inhibitory neurons are the medium spiny neurons, and the telencephalon inhibitory interneurons correspond to the cortical interneurons. But in addition, we found some evidence that other neurons might be implicated, although it could be that it's due to shared core neuronal mechanism. So Parkinson's disease was very different, and what we found is at the top are cholinergic and monoaminergic neurons, and this is what we were expecting because this is a broad category of neurons that include the dopaminergic neurons that are lost in the disease. But in addition, we found oligodendrocyte, which was unexpected, enteric neurons, as well as hindbrain neurons, and peptidergic neurons. So these results are interesting in light of Braak's hypothesis. So Braak is a neurologist who has studied a lot of postmortem Parkinson's samples, and he found lesions all over the nervous system, and he made this hypothesis that Parkinson's disease could start in the gut, where enteric neurons would come into contact with microbial product, which would lead to aggregation of alpha-synuclein. This would propagate along the vagus nerve to the brainstem and ultimately the substantia nigra. And so we found evidence for enteric neurons, for neurons located in the brainstem. So these are hindbrain neurons that were significant using L-score regression, but also some of the monoaminergic and cholinergic neurons are located in the brainstem, as well as neurons located in the substantia nigra. So our results are consistent with Braak's hypothesis. And finally, we could get our hands into case-control gene expression differences for Parkinson's disease, and we wanted to see if the cell type that we had found to be that we associated using GEX were also also implicated at a functional level in case control studies of Parkinson's disease. So we looked at whether genes that are upregulated or downregulated in people affected by Parkinson's disease in their substantia nigra, so these are post-mortem studies, were enriched in specific cell types from the midbrain. And so we found that upregulated genes were specifically enriched in oligodendrocytes, providing evidence that those cell types are indeed dysregulated in Parkinson's disease. And we also found that the dopaminergic neurons and other midbrain neurons, while downregulated genes were enriched in dopaminergic neurons and other types of neurons from the midbrain. And this is what we would expect because those neurons are lost, and so all genes that are expressed in those cell types are should be downregulated in patients. So one thing that was really nice, I think, was that we could get case control data at different stages of Parkinson's development. And what we could see is that in the early stage of Parkinson's development, the upregulated genes were enriched in oligodendrocytes, but there was no enrichment of downregulated genes in dopaminergic neurons, which was not the case when you look at later stage of Parkinson's disease. So this suggests that the oligodendrocytes are affected in the early stage of Parkinson's disease before the loss of dopaminergic neurons. So in conclusions, we could find that schizophrenia and other psychiatric disorders are in general associated with projecting excitatory neurons and projecting inhibitory neurons. That in contrast, Parkinson's disease has a very different association profile with association with cholinergic and monoaminergic neurons, oligodendrocytes and enteric neurons, and that this is providing new evidence for the hypothesis that Braak made, which is that Parkinson's could start in the gut. And we found alteration in oligodendrocyte and dopaminergic neurons, and oligodendrocyte already early in the disease. And so if you want to read more, the paper was just published this week, and you can find it on the Nature Genetics website. So, and of course, this work would not have been possible without all the different collaborators, and I'm ready for question if there's time.